If you've ever tried to balance a pole in your hand, you probably discovered it's pretty tricky, but the more you practice, the easier it gets. Hi, my name is Dylan Bates, and I'm using machine learning to make a computer teach itself how to balance a pole. Machine learning, a subset of AI, can often seem like magic. Through the different techniques of machine learning, computers can now do incredible things like diagnose cancer patients more accurately than doctors, play video games better than professionals, and generate works of art in the style of Mozart, Shakespeare, or Van Gogh. But it isn't magic, it's just math. In each of these applications, all that's happening is a computer is using a specific algorithm to minimize some function that could have thousands, millions, or even billions of parameters. We can harness the fact that all of these take place on a computer to speed up the development of control systems that can be implemented in the real world in applications like robotics or self-driving cars. So what does this have to do with balancing a pole? Well, let's imagine our setup as a cart with a pole on top. Using some of the same techniques from machine learning, we can input things that we know about the state of the pole, like its position, angle, and velocity, and it will output its best guess of how much the cart needs to move left or right in order to keep the pole balanced. We can do this by using a reward function. You can think of this like giving the computer some reward, like an M&M, for every fraction of a second it's able to keep the pole balanced. At this point, we can run a simulation until the pole falls, then use our algorithm to update all our parameters so the pole is more likely to balance in the future. Then, just repeat this over and over and over again, maximizing the number of virtual M&Ms it gets until it learns how to balance on its own. Ultimately, we can use the same process to train a self-driving car to drive in a simulation, letting it experience hazardous conditions like snow or near misses without putting the driver or the $100,000 car at risk. What makes my research unique is we can do this process entirely on a computer, harnessing the power of graphics cards to run hundreds or thousands of trials in minutes instead of hours. Once it's done, once it's taught itself how to balance a virtual pole or drive a virtual car, we can use the same function it learned to balance a pole or drive a car in the real world. The idea of using machine learning to balance a pole is just a proof of concept that can make other machine learning applications more efficient, letting computers do all the work before implementing what they learned in real life. Large, complex systems where data is dangerous, expensive, or difficult to obtain, like cars, satellites, or even reusable rockets. All of these could benefit from this cheaper, faster, computer-assisted process. Thank you.
For centuries, civilizations have been in conflict with one another for survival, resources, and economic prosperity. In this way, we are not much different from bacteria. As we engage in conflict above ground and in plain view, a hidden war transpires below ground and is costing us on a global scale over $1 billion annually. At the center of this phenomenon is Ralstonia solanacearum, a plant pathogenic bacterium that limits global crop production and is a causal agent of bacterial wilt disease in tomato and brown rot in potato. This bacteria affects other major crops, such as eggplants, peppers, tobacco, and bananas. Imagine your salsa and spaghetti sauce without freshly chopped tomatoes, your hamburgers without a side of your favorite crispy fries, or your baba ganoush without roasted eggplants. This disease affects countries worldwide and is particularly devastating in the tropics, where a large percentage of the food insecure population lives and depends on agriculture for sustenance. This pathogen can have serious economic ramifications for growers, as once established in the field, it is difficult to eradicate, leaving farmers no choice but to abandon that land completely. Currently available disease management strategies have limited success, and some are dangerous to the environment. So what is the solution to this microscopic threat that impacts our survival, resources, and prosperity? Well, why not use their own weapons against them? Ralstonia competes for resources against closely related groups of bacteria by producing and injecting toxic proteins to slow or kill its competitors. What is amazing about these toxins is that they can precisely target a bacterial cell type. By understanding how bacteria compete, we can engineer and develop environmentally safe tactics to manage Ralstonia and use their own weapons against them. Towards this end, I'm studying how these toxins function in Ralstonia. To date, I have found that there are some cells that carry 2 toxins, while other cells carry 13 or more. The diversity in how these toxins function vary greatly. Some break down competitor cell DNA, while others probe holes into cell wall membranes. In other words, some kill from the outside in, while others kill from the inside out. The question now is, how can we engineer these toxins for our benefit? To answer this question, I'm looking into individual toxin genes in Ralstonia and identifying their modes of action, method of delivery, environmental viability, and their competitor profiles. In doing so, we can identify the best toxin or toxins to use to specifically target bacterial populations and halt their march before reaching the host plant, preventing the colonization of the host plant and the development of disease, hence mitigating agricultural loss, economic impact, and most importantly, the dishes we love to eat.
No one likes flooding. Whether it's happened in your basement or your backyard before, typically nothing good comes from it. Throughout the years, we've seen the extreme detrimental impacts from floodwaters, mainly brought about by heavy rainfall, hurricanes, or dam failures. Heavy rainfall and river flooding are top indicators of global climate change. And unfortunately, as the climate continues to change, we can expect to experience more frequent and more intense storms, resulting in more flooding. Here in North Carolina each year, it is a common sight, especially in the coastal plain, to see homes, businesses, and farms devastated by floodwaters, mainly brought about by hurricanes. Hurricane Florence in 2018 and Hurricane Matthew in 2016 hit the same agricultural communities back to back, taking 85 human lives and causing $17.6 billion in losses, much of which were agricultural crops and livestock. These communities can no longer afford to experience the repercussions from flooding. So what do? What if we could farm the floodwaters? Farming is about knowing the lay of the land and working with the land and livestock to make the most out of what we can. What if I told you that there is a method that could redefine the concepts of farming? The method is called water farming, and my research hypothesizes that it is a solution for mitigating our future floodwaters. Water farming uses natural land features as opposed to traditional human-engineered systems that are made from concrete, such as dams or levees. Traditional human-engineered systems are highly hazardous, outdated, and have failed to function when needed the most. My research suggests that nature-based practices are a more sustainable approach to flood management. So you're probably wondering, what does a natural defense system to flood management look like? Well, there are a variety of approaches, such as restoring wetlands, planting forested areas, expanding floodplains. Nature-based solutions like forests and wetlands can slow down water from storms and store water for an extended period of time. In fact, I have found that water flow could be reduced by more than 30% at downstream if nature-based practices are employed. My research focuses on the top and most effective water farming practices for individual landowners and agricultural communities in eastern North Carolina. We have coined our line of work Floodwise, and our program ensures landowners with the educational tools and financial incentives for adopting nature-based practices on their properties. Dear audience, flooding and storms will continue, and we need to be prepared. We must take precautionary measures and invest in Floodwise. Thank you.
A few years back, I attended a community meeting about an upcoming street improvement in our neighborhood. I had so many things to share, but I didn't get enough time because of my language barriers. One of the community leaders even couldn't attend due to some family emergencies. But not having enough time or an emergency or anything like that shouldn't prevent us from participating in matters that would impact our communities. Fortunately, and with the help of digital technologies such as online surveys or virtual meetings, we now have the opportunity to fully express our ideas anytime and from anywhere. Well, not really. Regardless of the meeting being in person or virtual, many participants would have a hard time understanding the scale or the location of future projects just by seeing their maps on the wall or beautiful renderings on computer screens. What if there was a technology that would let you envision the future design right in its location and even before it's built? The good news is the technology is out there. Do you remember a couple of years ago, people were all around the place with their smartphones catching Pokemons that did not physically exist? They use a technology called augmented reality that superimposes digital data, whatever it might be, a Pokemon, an image, or I would say 3D models of future designs, into the real world. For my PhD, I've developed a platform equipped with augmented reality to enable community members view the design overlaid on their surrounding environment and even interact with it. They can add missing features, manipulate them instantly, vote on their preferred design, and share their ideas with fellow residents and decision makers on the go and from the palm of their hands. Smartphones are now everywhere. More than 81% of Americans now own a smartphone. For 26% of lower income Americans, smartphones are the only way to interact with the digital world. More importantly, a smartphone is the only digital technology that is equally owned by different racial and ethnic groups. I've been working with a group of communities of color in North Carolina that have inequitable access to street elements such as sidewalk that make neighborhoods healthy and safe. I've implemented and assessed my platform and found that augmented reality is a great addition to current community engagement methods. It improves participation by letting people be in the actual space and discover things they didn't notice on paper. My hope is that by using my platform, we can achieve a more accessible, a more intuitive, and beyond that, a more inclusive community engagement, so everyone can participate in making their neighborhoods better places to live. Thank you.
Hello everyone, let me start with a question. How many of you are currently wearing a dress or sitting near a textile product that is printed? If you find it difficult to identify a printed product, look at both sides of the fabrics. Printed products only contain coloration on technical phase or design side. Well, there are a few exceptions, like flags and scarves. They contain coloration on both sides. Chances are you all own some printed products, whether apparel or home furnishing. Textile printing is a 150 billion market and a popular market trend. Here are some facts regarding printing. It requires 6 to 12 weeks to print with traditional techniques. Chemical and energy costs are very high. As much as 60 liters of water is used to print 1 meter fabric. Moreover, excess dyes and chemicals may enter waterways from improper handling. Traditional printing can be performed in various techniques, such as blocks, flat screen, or rotary screen. In my research, I am investigating an emerging printing technology, digital printing, and comparing it to rotary printing, which is the most predominant traditional printing process. To do so, I have developed a comprehensive survey instrument to collect industrial data on associated costs and environmental impacts. Textile industry has started to embrace digital printing for its ability to print with increased number of colors and greater print quality. It is believed to be a fast and environment-friendly process. However, these advantages have not yet been quantified properly. With my analysis, companies will be able to take more informed decisions by identifying cost burdens and key improvement areas. Initial data reveals that digital printing utilizes 1/5 amount of water, 30% less energy, and only 10% chemicals compared to conventional ones. As I collect and analyze more data, I am hoping to identify the fastest and most cost-efficient sustainable printing process. This might will encourage manufacturers to choose printing process wisely. So guys, next time during shopping, think about how your colorful printed item may improve in future both in quality and complexity in design, as well as sustainability of the coloration process. And remember, NC State is helping to improve this process. Thank you.
Imagine you step outside and you're suddenly ankle deep in sweat. It stings and you're angry. Why would this be happening? Swell backups happen when gigantic solid deposits accumulate inside of swell lines that ends up clogging the pipes. But the question is, how do these solid deposits get there? You'll be surprised to hear it, but we are responsible for this. Let me ask you, what do you do with excess fat, oil and grease after cooking? Many people pour it down the kitchen sink where it ends up in the swell system. There, it reacts with calcium to form the swell giant known as fog deposits. In the United States, around 2 billion gallons of untreated wastewater are released annually because of fog deposits, costing $25 billion of taxpayers' dollars to clean up. So don't be surprised if your basement or street is flooded with sewage one day. But don't get frightened as well, because I have a solution. Fog deposit formation requires two key ingredients, fat, oil and grease, and calcium. Calcium comes from the cement used in swell line construction. Cement has two calcium components, calcium hydroxide, where the calcium is loosely bonded and can easily release calcium. The other one is calcium silicate hydrates, where the calcium is strongly bonded. To minimize fog deposit formation, I am replacing a percentage of cement with fly ash, because fly ash has two major benefits. It is very low in calcium content and it can react with cement to convert calcium hydroxide to calcium silicate hydrates that does not release calcium readily. But where can I find fly ash? Fly ash is a byproduct of coal-fired power plants, and power plant companies are looking for safer ways to get rid of it. I found that 50% fly ash replacement can reduce fog deposit formation by 55%. When I started this research, my aim was to reduce swell giants. But as I progressed through this, I realized my work can benefit in multiple ways. It can reduce fog deposit formation and save a chunk of taxpayers' money. It can use fly ash and reduce water waste contamination. And finally, it can save a lot of money by reducing soil and construction cost. So, I am doing my part to fight swell giants, and you can too by not pouring fat, oil and grease down the kitchen sink. Thank you.
Reduce, reuse, recycle. You know the symbol. It's been around since the first Earth Day in 1970 and has been drilled in our schools and PSAs in the decades since. Today, going green is all the rage. But what comes to mind when you think about recycling? Likely the separation of bottles and cans and cardboard and putting them in their respective bins. But what about the recycling that's going on all around us? It's in the soil, it's in the water, it's in your intestines, and it's all thanks to microbes. So why don't we harness their power for our recycling? That's exactly what my research does. It focuses on the repurpose and reuse of a small chemical, isobutylene, seen in the middle of my slide. Isobutylene is used in the production of plastics and rubbers and cosmetics and fuels, and not a day goes by that you don't interact with something that started from this chemical. In fact, it's in such high demand that you can count it the same way you did playing tag. 1,000 pounds, 1,000 pounds. Now, while isobutylene itself isn't that harmful, its natural byproducts are. So let's get to recycling. A lab I work in found on this very campus a microbe that can eat isobutylene. And while doing so, it actually produces more industrially relevant chemicals. There it is again. Reduce, reuse, recycle. We reduce the toxic byproduct, reuse the original chemical, and recycle it into other important chemicals. But it's not enough to know that it can do it, it's important to know how. So I dove deep and determined the entire genetic makeup of this organism. From those 6.5 million A's, T's, C's, and G's, I was able to determine the genes responsible for isobutylene metabolism. I even produced one individually to prove that it can work in vitro, without microbes present. And while doing so, I determined that this thing actually works on other chemicals as well. So it's not a stretch to imagine that if I mess with this or add other genes, we can actually take care of other harmful chemicals too. What's more, this whole system seems to be controlled on something that's akin to a dimmer switch. The more recyclable material that's there, the harder and faster and stronger it works. So next time you're holding a bottle or that styrofoam takeout, don't just think about recycling those products. Remember, the world around you is recycling too, and we can make it better. All we have to do is look at the mechanisms behind how nature itself can reduce, reuse, and recycle. Thank you.
Have you ever considered that part of your story may be written before you're even born due to the DNA you inherit? Let's consider a young girl named Evelyn who was born with spinal muscular atrophy. This debilitating disease will likely leave Evelyn unable to breathe on her own simply because she inherited a genetic mutation. Evelyn, however, was able to rewrite her story due to a revolutionary gene therapy treatment that introduces copies of the DNA that are missing from her genome. Because of this treatment, Evelyn today is a healthy, thriving 6-year-old. Her story is truly inspiring. However, her treatment was one of the most expensive drug therapies ever, costing over $2 million. At this high price, gene therapy is simply not accessible to the majority of patients. So why does gene therapy cost so much? The main driver is a lack of standardized manufacturing. In their infancy, automobiles too were plagued by high costs for similar reasons. However, over the last 100 years, the price of a car has fallen significantly due to standardized manufacturing, which was first implemented by Henry Ford in his idea of the assembly line. Think about it. Today, almost every family can afford a car. Likewise, the accessibility of gene therapy treatments will continue to improve as standardized manufacturing is implemented. Gene therapy treatments rely on the use of a viral-based delivery vehicle to transfer DNA to patients. Multiple vehicle variants exist, each with their own pros and cons. All vehicles are produced by special cell lines and bioreactors. Process-related impurities must be removed prior to injection into humans. This is generally accomplished by flowing the reactor's contents over a capture surface that is functionalized with an engineered protein that can bind the delivery vehicle while the impurities flow through as waste. While this capture step seems straightforward to implement in a standardized fashion, it is complicated by the fact that each type of delivery vehicle currently requires its own unique capture agent for purification. This limitation impedes standardized manufacturing across different gene therapy products. When considering the automobile, Henry Ford took great strides to simplify manufacturing. He went as far as only producing the Model T in one color to save time and money. In a similar vein, my research has focused on simplifying one critical piece of gene therapy production. I'm trying to identify a single capture agent that can be used to purify the most common DNA delivery vehicles. Throughout my PhD, I developed tools to engineer proteins with novel binding activity. I've applied these tools to generate proteins that can bind conserved regions between common delivery vehicles. In turn, these engineered binding proteins can be used as a universal capture agent to standardize manufacturing. Just as Henry Ford standardized the production of the automobile, I am standardizing the production of gene therapy products.
Have you ever been stuck in traffic? And maybe you thought that there were too many cars, or too many people, or that whoever designed the road network didn't think about so many individuals flowing at the same time from one side of the city to the other. Whenever there is a movement of many individuals, the network in which they move is a key factor for a smooth, uninterrupted flow. In a similar way, a battery separator provides the road network for the lithium ions to flow from one electrode to the other. The battery separator is the only thing preventing the electrodes to come in contact and to develop an electrical short inside the cell. The FAA reports that on average, a battery fire on a plane occurs every 10 days. Reports of exploding cell phones have been published in newspapers, and batteries from Apple, HP, Toshiba, Lenovo, Dell and other manufacturers were recalled due to fires and explosions. A battery separator is only 1/1000th of an inch thick, yet is the only safety measure to prevent your cell phone, laptop or car battery from catching on fire. In the past 5 years, I worked to understand and design the ideal network to improve the safety and stability of lithium-ion battery separators by simply using fibers, just like the ones that make up your clothes. The idea was to create membranes with an existing technology, but to extend it to an application that was never explored before. First, we use a process called melt blowing, which is commonly used to make wipes, filters or even face masks, by simply ejecting fibers into air to create inexpensive, thermally stable and efficient membranes. In my research, I found out that not only these membranes work as battery separators, but that a specific combination of microfibers and nanofibers improve the stability of the battery. The microfibers gives the scaffolding with high strength, while the nanofibers drape over the microfibers to create the ideal network for the lithium ions. But I didn't stop there. To prove that this was indeed the ideal network, I used a different process, and this time the fibers were ejected into a turbulent liquid flow. Well, it turns out that even with these new separators, the same combination of micropores and nanopores gives the highest stability and the best performance. Just like the combination of highways and residential streets reduces the traffic congestion in our cities. Could you imagine living in a city where there are only highways or only residential streets? Traffic would be a nightmare. By creating the ideal network for the lithium ions, these specific separator structures could improve the stability of next generation lithium ion batteries, decrease the chance of failures, and keep you safe from battery fires. Thank you.
When I was a kid, there were two things that I really disliked doing: going to the dentist and chores, especially cleaning the bathroom. Now, as an adult, I recognize the importance of going to the dentist and cleaning your bathroom. But I also realize that these two activities share one main thing in common, with a key component of my research: biofilms. Biofilms are a surface-attached community of bacteria that make a gel containing proteins, sugars, and DNA in order to protect themselves from antibiotics and the environment around them. Now, dental plaques are a type of biofilm made from the bacteria inside your mouth. And that pink stuff inside your sinks and showers at home, that's another biofilm made from another bacteria. Now, these biofilms are much more common and much less harmful, especially compared to a biofilm infection on a hip implant. Because biofilms increase bacterial survivability on surfaces such as medical implants, hospital equipment, and clothing, they are implicated in as many as 80% of human bacterial infections, ranging from endocarditis to cystic fibrosis. In addition, bacteria inside of the biofilm are 10 to 1,000 times more resistant against antibiotics relative to a single bacterium. So, if biofilms are rendering our antibiotics ineffective, what can we do? Currently, the FDA has no approved drug for the treatment of a bacterial biofilm. However, our research group was interested in taking on this challenge. We were inspired by a small class of molecules isolated from sea sponges called synoxazolidinones. So I went out and synthesized a whole new set of synoxazolidinone derivatives in order to evaluate their properties against methicillin-resistant staph, or MRSA, biofilms. To our delight, we identified several molecules that were able to successfully inhibit the formation of MRSA biofilms, as well as break up or disperse preformed MRSA biofilms. Going one step further, we collaborated with our colleagues at the College of Veterinary Medicine and we were able to confirm that our molecules significantly reduced the amount of living bacteria inside of a much more robust and realistic biofilm model. Finally, we were able to show that our molecules, in combination with FDA-approved antibiotics, significantly increased the efficacy of the antibiotic 8-fold using only a small amount of our molecule. At the end of the day, we have a very long road ahead of us before we deliver the world's first biofilm treatment. However, we have shown with our research that these synoxazole derivatives are a great starting point for the development of an anti-immersive biofilm agent.